Well, thank you very much for the invitation, Ulrich. It's a pleasure to be there, at least virtually. I would prefer to be there in person, but as it is, this is good. I'm going to hopefully take things in a slightly different direction than Dr. Wooster has done, but hopefully this will be complementary as well and talk about some of the individual toxin families and some of the effects that they have on human envenomations in particular. So to do this, we'll take a look at the snakes themselves a little bit, the venom delivery systems that we see across venomous reptiles, talk a little bit about venom composition, and then focus in on a couple of protein families that arguably are amongst the most important ones. We'll first look at systems that are targeted by these various toxins, and then we'll look at the toxins themselves in a little more detail. And then towards the end, I'll try to wrap things up and provide some take-home points for you as well. Well, as we've seen, World Health Organization has in fact recognized human envenomations as a neglected tropical disease, on several different occasions. And hopefully this global strategy will in fact be met by 2030. But if we think about envenomation in humans, this is a very complex and obviously serious medical emergency that is constantly evolving itself and requires all sorts of modifications along the way and attention to details as far as patient care. And as Wolfgang mentioned, we can see very significant differences between species and sometimes even within a single species across its range. And we'll talk a little bit about that. So the symptoms that we see produced in snake envenomations are a direct result of the particular biochemistry of the venom, but we also want to keep in mind that the details of the human that's envenomated and any kind of comorbidities or any other conditions like that can also be very, very effective and important on determining what is going to happen, but also the outcome of a bite. So in rural areas, which is, of course, much of the world that is suffering from snake bite problems, getting to treatment in a timely fashion is a major, major problem. And of course, time is important for ameliorating serious effects of envenomation. I'm going to go kind of mention one point that is rather annoying to me right now, because in the United States, we've got basically only 2 antivenoms that are anti-pit viper antivenoms. And so there is a kind of cavalier attitude amongst a lot of physicians that species identification is not important. But I'd argue that it is important, that we need to, in fact, identify the species. And in the United States, only about half of the bites are actually identified 2 species, which is kind of surprising to me. Well, one example I'm going to mention is from a paper that Kara Smith published recently titled Snakes on a Plain, that is the Great Plains of the United States. And we'll look at that in a moment here. So why do we want to know? Well, even in the United States, where we don't have the diversity as we do in some tropical countries around the globe, we have expectations of a bite depending on the type of venom and what we call type 1 versus type 2 venoms relating to a variety of different species of rattlesnakes and other vipers for that matter, can be very, very different. So Western diamondbacks, such as this one here, produce a lot of local damage. They can, in fact, go systemic and cause problems, but that's typically a later manifestation. In Mojave rattlesnakes that we've heard a little bit about already, what we would expect are less in the way of local effects, but in fact, development of a variety of problems, including neurotoxic manifestations. But that doesn't actually seem to be the case, at least in the few studies that have looked at human envenomations in a systematic sort of fashion. We see geographic variation in venom composition, as we've heard just now from Wolfgang. And I want to mention one that's a little bit closer to home for me, and that's the prairie rattlesnake. And this is a snake that its range extends all the way from southern Canada down into northern Mexico. And recently we sampled this, and right at this red line here, which corresponds roughly to Interstate 70, if you know anything about highways in America. The northern and the southern phenotypes of the prairie rattlesnake are very, very different. And in fact, they follow this pattern where there's this disjunct shift right at about that line. And this is just north of Denver, Colorado, the capital city of Colorado. And in the northern phenotype, we find a huge amount of myotoxin, sometimes 40% or more of the venom comprised of this one toxin. Whereas in the southern regions, what we see is a snake venom metalloproteinase rich phenotype. And so again, we've got this very, very disjunct and distinct shift in overall composition. Well, a recent paper published by Chema Gutierrez out of Costa Rica has really argued for using an interdisciplinary research approach towards trying to deal with this problem of snakebite. And so today what we're going to do is look at a bit of the ecology of snakes, relate that in gross kind of broad brush treatment, and hopefully I won't violate Wolfgang's caveat of oversimplification here. But we'll look at venom composition and toxicity and trends that we see in snakes, and then how that relates to clinical aspects of envenomation. Well, if we think about venomous snakes globally, what we see is a wide variety of different morphologies and patterns, etc. So we've got elapid snakes shown here, a couple of different vipers here, and then on the bottom row here, some of the rear fang snakes, colubrid snakes that for the most part don't present much in the way of a human risk, but they're very interesting biologically. We also have a few venomous lizards. Heloderma has 2 or 3 or perhaps 4 described species, depending. The Gila monster that extends from the southwestern United States into Mexico, and then a more Mexican and Central American species, the beaded lizard. Very different sort of delivery system here, with venom glands on the lower jaw here associated with grooved teeth, and also these have a typically bulldog type of bite. Rear fang snakes actually produce interesting venoms, and we'll come back to this particular species, the Amazon puffing snake, but they have a very obviously serous secretory gland that's associated with enlarged teeth on the back of the maxillary. And in fact, in some of these species, this is deeply grooved, and so they can, in fact, deliver venom at a moderate rate, but typically require a longer hold time for human envenomations to develop anything significant. Elapids and viperids, of course, very different sort of situation, where we have fixed fangs on a maxillary bone that's elongated in typical elapids. And then associated with that is a main gland here, leading through an accessory gland and out to the fang itself. We've worked extensively with a variety of species of rattlesnakes, and so part of the bias of this talk will be, in fact, towards rattlesnakes. But in rattlesnakes, we have a rather interesting mechanism for delivering venom very deeply into the tissues of prey. The fangs can be quite elongate in viperids, and they have this typical bevel opening, very much like a syringe needle tip. The main gland has associated with it a very potent adductor muscle that will deliver a large amount of venom rapidly. And so, in fact, they represent a significant health risk to humans because of the large volume they can deliver very rapidly. Okay. If you think about venom production in pit vipers, and this is largely the case for the few elapids that have been looked at as well, when one extracts venom, you actually stimulate the parasympathetic nervous system into gearing up the cells, that is the secretory cells, in the production of proteins. All sorts of genes are turned on. Venom proteins are produced in large amounts. They're formed into vesicles, which then are exocytosed. And in the front fang snakes, we have then a fair amount of venom that is stored ready to go at a moment's notice in the ductules, that are these tubular looking areas in the gland. And so we have then venom is replenished. This takes somewhere between 10 days and 2 weeks, depending on species. And of course, this is a highly regulated process, and it turns out, just like the venom components are usurped regulatory components from the systems, the regulation of transcription and translation of these interesting proteins is in fact usurping already well-known systems like ERK and MAP kinase. So if we think about the venoms themselves, and this is produced from the snake's fang, what we see is mostly proteins and peptides. There are a bunch of small molecules that really need to have more attention turned in their direction, but these are co-opted regulatory compounds. They're very, very potent. And if we look at a typical venom, the number of proteins families represented is relatively small, but a common pattern that we see is that within a single protein family, and if you remember Dr. Wooster's, Wolfgang's representative of the snake venom metalloproteinases, what we see is that in a single protein family, there are many, many different genes, orthologs that are present, and many of these homologs are actually produced. Typically, these proteins are also very, very stable, and they have a stable molecular scaffold. Well, in order to study the venoms, the first thing one needs to do is, in fact, get the venoms. Some of these are very easily obtained. Vipers have a large stored amount of venom in their glands, and so extraction is relatively straightforward. Some of the rear fang snakes require a bit more intervention, as it were, with a drug regimen to get them to secrete significant amounts. It's become rather old school now, but I like to use SDS-Page as a first look, because we actually know quite a bit about things like rattlesnake venom composition. And from the position on the gels, we can actually predict the most probable protein families that are expressed. The other thing is that even starting students in the lab, undergraduates, can run these gels and get interesting results quite readily. What we see is that in some of these cases, we see very prominent appearance of some of the bands, such as the snake venom metalloproteinases P3 class, and again here P1 class of metalloproteinases, whereas in many of the other venoms, they're lacking those, or it's in very, very low concentration. So we can get a fair amount of information using this simple technique. Well, proteomics, of course, has followed the omics trend in becoming very, very prominent. And so Juan Calvetti out of Valencia, Spain, has in fact championed the use of snake venomics for trying to understand venom composition. And one of the great things is that it requires very little material. And we have the potential then to catalog an entire proteome for a species. If we couple that with transcriptomic studies, then we can get even more detailed information, because we have the potential to sequence all of the mRNA transcripts that are present in a particular species gland and get an idea of what the potential is there for that species. And that can lead us to a complete catalog of toxins for any given species. And so here's an example of some work that Cassie Modal did a number of years ago in my lab with the Amazon puffing snake. And so on the left is the transcriptome, on the right, the proteome. And as we heard a little bit ago, there can be a very significant difference between the transcriptome, the potential of what can be produced, and the proteome, what actually is produced by the snake. So finally, then, genomic studies are making inroads into toxinology of snake venoms. And in this context, we can really look at the entire genome and look at venoms and toxins that are expressed in that context. In addition, we can start talking about aspects of gene regulation, control, and variation of expression patterns. And this study that was published in Todd Casto's lab has actually mapped the majority of toxin families to particular chromosomes in the prairie rattlesnake, as shown here. And so the interesting thing about this is that many of the dominant protein families are found in microchromosomes, with a couple of outliers here, in particular myotoxin A protamine homologs. So we have a number of different tools then at our disposal for looking at venoms and venom composition in a lot of detail. And so again, not wanting to oversimplify, if we look at a variety of different snakes, we can make some generalizations. And this is looking at dominance of protein families. And what we see in most of the vipers is that there's a predominance of enzyme type of toxins, and those are shown by these cooler colors here. So this is characteristic of what we call the type 1 venoms. They're lower toxicity but higher enzymatic activity, in particular metalloproteinases. And that contrasts with type 2 venoms. And what we see in Crotalus scutellatus, the Mojave rattlesnake, is they can't really make up their mind which ones to express. And so some populations show this type 1 or B type of phenotype, and others show a type 2. There are a couple other species that have characteristically very, very potent venoms. Tiger rattlesnakes, also found in southern Arizona, and then tropical rattlesnakes have a high preponderance of these beta neurotoxic phospholipase A2-based toxins. If we do the same kind of broad brush on front fang snakes of the family Elapidae, we see a very different sort of pattern. And again, the hot colors here are indicating three-finger toxins. Now, there are a bunch of different activities associated with three-finger toxins, but in general, elapids have invested very heavily in three-finger toxins and, in some cases, phospholipase-based toxins. Finally, then, just to round things out here, if we look at rear fang snakes, we don't have as much information, but there's a growing body of evidence on composition here, as well as some of the interesting effects that they can have. And so we see some that fall into what we would consider more of a type 1 versus a type 2 sort of pattern, where we have in the type 2 a dominance of highly toxic components, in particular these three-finger toxins. But these are often taxon specific. That is, they show very low toxicity towards mammals typically, but high toxicity towards lizards and birds. So there's some interesting patterns going on in colubrids, but because colubrids, other than something like the boomslang shown here, don't really contribute significantly to human morbidity and mortality from their bites, we're not going to really talk about them much more. Well, if we think about the systems that are targeted by these toxins, there are a lot of different systems. And if you think about it, it makes perfect sense to target the peripheral nervous system and the neuromuscular junction in particular, because if you can incapacitate this communication system, then you have, in fact, immobilized your would-be prey. We have toxins that are hitting at the presynaptic site here, and these are characterized by a lot of the beta neurotoxins, those phospholipase-based 2 subunit toxins that are moderately common in some rattlesnake venoms, and then there are some similar types of toxins, multimeric in many cases, in some of the elapids. On the other side of the cleft, we can have postsynaptic inhibitors, and these are often alpha neurotoxins in the three-finger toxin family, and characteristic of the very potent toxins of many elapid venoms. But we also see three-finger toxins in a number of the colubrid snakes. If we look at the muscular system, in addition to targeting the neuromuscular junction, there are a variety of toxins, again, phospholipase-based, that act as myotoxins to disrupt the actual physical structure of the muscles themselves. One that we've worked with a bit here, myotoxin A, which is a homologue of crotamine found in South American rattlesnakes, is apparently incapacitating calcium pumps in the sarcoplasmic reticulum. And this is a very potent toxin that we'll see in a little bit. But when we think about systems, it's really the vasculature and the coagulation cascade systems that have been really targeted by a wide variety of different toxin families found in venomous snakes. And so snake venom metalloproteinases result in structural, or can result in structural damage. We see these characteristic of vipers, some elapids, many colubrids as well. Hemolysis generally is due to action of phospholipases, not an overly toxic activity, but a very common one. PLA2s are found in most species of venomous snakes. We have some specialized snake venom serine proteinases that are kallikrein-like that can liberate bradykinin, leading to circulatory collapse and perhaps induction of rapid shock. We have a variety of components, disintegrins and C-type lectins, that can affect platelets. Some of these promote, some inhibit aggregation of platelets. There are thrombin-like serine proteinases in the venoms, very common in many of the vipers, less common in the elapids that can lead to a depletion of fibrinogen and interfere then very significantly with the capacity to clot. So we have a 1-2 punch here. If we're breaking open capillaries and blood vessels, and then we're inhibiting the capacity of the repair mechanisms, that's going to be a problem. There's some specialized toxins that are factor 10 activators, and these are found in things like a few of the lapids, as well as the boomslang, and very problematic in all of those species. So we're going to take a look at the flip side now and talk about some of the venom toxin families. And again, this is looking at a couple of them, not all the different ones that are found, but arguably these 5 families are amongst the most important ones as far as producing clinical signs in humans. Metalloproteinases are very, very common in a variety of venoms, in particular viperids and colubrids. They're functionally important for a number of different reasons. They result in protein degradation, hemorrhage. They may be associated with a biological role of degradation of the tissues of prey even before the snake has swallowed it. In human envenomations, they lead to a variety of local and later systemic changes that can be very, very problematic. And these often don't respond very well to antivenom therapies, especially if it's delayed for some period of time. There tends to be an inverse relationship between the most toxic, as far as lethal toxicity, venoms, and those that have high metalloproteinase activity. And this is what led us to this type 1, type 2 dichotomy initially. In the metalloproteinases, we see that it has a modular sort of design in the way these molecules are put together. And the P3 have all 3, P1 only have the metalloproteinase domains. And so it's only been looked at by a few researchers, primarily in Brazil, that have suggested that there are different substrates and therefore differential toxicities for these different families of metalloproteinases. Regardless, they're very abundant components in many times. And locally, in the US at least, there is this adage, time is tissue. And in fact, that is related to the point that these, along with PLA2s, can lead to very severe necrosis, ecchymosis, hemorrhage, etc., that then doesn't respond well to antivenom therapy. Serine proteinases are primarily targeting aspects of the coagulation cascade. And we have a number of these that are that have the capacity to release very potent peptide signals that result in circulatory system relaxation and collapse of blood pressure. And interestingly, they can be extremely abundant in venom. So a Massasauga rattlesnake that we have here in Colorado has at least 10 to 12 homologs that are found in the venom itself. And for that matter, prairie rattlesnakes in our area have a similar number of what looked initially to be redundant expressions of the same serine proteinases, but they have subtle differences in their specificities. One of the things that can happen from envenomation by a snake like the midget-faded rattlesnake is that coagulation is disrupted. And in particular, if one follows fibrinogen levels here over time, initially, this individual presented with uncoagulable blood. And then after significant amounts of antivenom, fibrinogen levels began to return to normal levels here. So that can be one of the effects of the thrombin-like serine proteinases that are in some of these venoms. In our area, we have prairie rattlesnakes that produce this small toxin, myotoxin A. It's only 42 amino acids in overall length, but it can account for an extremely large amount of the total venom. When prey are bitten by snakes containing this large amount of myotoxin A, they show almost instantaneous tetanic paralysis. So rather than the flaccid paralysis of the neurotoxins that we mentioned a little bit ago, this results in tetanic paralysis, largely, we believe, by its effects on the sarcoplasmic reticulum calcium pumps. Phospholipases, as we mentioned, are in fact broadly distributed across many venomous reptiles, and they can be either monomeric enzymes or they can in fact form multimeric toxins, and those tend to be highly lethal toxins. They're also found in things like beaded lizard and Gila monster venom. And so we see these in some of the rear fang colubrid snakes, such as this lyre snake here. But the elapids and the viperids almost all have phospholipase A2 of one sort or another. Some of these are actually potent neurotoxins, and these are the crotoxin homologs. Mojave rattlesnake in the United States is one of the most important problematic species as far as human bites, because it's common. It gets to be a moderate size. It tolerates human interventions and occurrence pretty well. And so, as Wolfgang showed a little bit ago, it can show variation in different areas and different populations. It occurs pretty broadly across the southwestern part of the United States and then extends very far down into Mexico. But we have a number of other species in the United States and in Mexico that produce homologs of these potent toxins. And some of these are characteristic of the venom, such as tigris and concolor, and others, some populations have them, some don't. So it's an interesting jigsaw puzzle of occurrence that's only been partially explained. As we go into Mexico, a wide variety of species also show the presence of these crotoxin homologs. Last family I want to mention are the three-finger toxins. And again, these are often the toxin family that gives things like the elapids their real punch, whether it's a terrestrial elapid such as Nyakuuthia or Laticata, a semi-aquatic marine snake. Also, we find them in some of the rear fang snakes, such as this boiga here. It's a particularly stable scaffold, and what we have seen among venomous snakes is that it has functionally diversified tremendously, and we have 10 or 12 different types of pharmacologies associated with it, including a very, very potent postsynaptic neurotoxic activity. And it can present major, major problems during human envenomations. In part, this is because it binds very, very well to the receptors. And so the alpha neurotoxins have evolved to recognize and bind to and incapacitate the acetylcholine receptors of skeletal muscle at a very, very low level. And so they are small molecules. They're very tightly tied together by disulfides. So they move through the systems fairly rapidly. And they can often be at fairly high concentrations. So cobratoxin, which is a classical alpha neurotoxin, can make up 15 to 20% of the total venom that's expressed in several species of the cobras. In an untreated scenario, this can in fact lead to fatal outcomes, oftentimes by phrenic nerve diaphragm incapacitation, and so a patient can no longer breathe. It's respiratory failure results. There are some new therapies that are in experimental phases right now, where in several labs are trying to come up with universal anti-three-finger toxin antibodies. And if these are successful, that would be quite a boon to treating this very, very problematic group and common family of toxins that are found in snake venoms. So there are some other therapies, and again, Wolfgang mentioned this in passing. Raspolidib is an inhibitor of secretory phospholipases, and it looks like this molecule, the small molecule, also has activity against not only the enzymatic versions of PLA2, but also some of these presynaptic neurotoxic PLA2s. Another compound, murimistat, is an inhibitor of snake venom metalloproteinases. Both of these small drugs seem to show a fair degree of promise, and several of them are undergoing clinical trials at present. And if they in fact prove to be useful for dealing with these very common families of snake venom proteins, then their utility as a first aid therapy is going to be absolutely tremendous. They're stable. They don't require cold chain storage that is typical of many antivenoms. They can be administered in the field by basically only lightly trained personnel, and so it doesn't require the intervention that administration of antibodies in the form of antivenoms typically would. Yeah. Well, What I've tried to do is give you an overview of some of the protein families that are most common in these different types of venoms. And they include enzymes as well as specific non-enzymatic toxins. Variation is kind of the name of the game. We've seen that in the previous presentation as well. And when we get human envenomations, we have a complex and importantly evolving medical crisis. A recent conference I went to that involved a lot of clinicians stressed the point that things are going to get better before they get worse, even if you're producing or giving antivenom. Again, this is resulting from the biochemistry of the venoms, the specifics of a particular individual and species, but the victim physiologically and comorbidity can also have a very important and largely unappreciated effect on this. When we think about how these toxins are working, again, we've got regulatory molecules that have been co-opted and overexpressed. They're produced and delivered at inappropriate times in huge quantities. And as a result of that delivery, all sorts of regulatory systems go haywire. And we have major, major problems leading to the varied symptoms that we see in human envenomations. Okay? On the other hand, we don't want to just point out the negative here. And a lot of these venoms actually have some really cool things going on with them. Some of these that are being developed as therapeutic drugs, and certainly as what I'd like to call molecular tweezers for understanding other systems. They have lots of potential there as well. As far as human envenomations, there's still a need for much better integration of basic and clinical research. And if these small molecule drugs do turn out to be first aid therapeutics, then in fact, we're going to see quite a change, hopefully, in the outcomes of even rural envenomations. Okay? I'd like to point out that where at all possible, it is very helpful to us to know what the species IDs are. And in fact, that will help us to understand levels of variation in a situation like a human envenomation. Okay. Well, to wrap things up, I just want to mention in passing, there is a Gordon Research Conference on this topic that will be happening next year in Portland, Maine. So look for that. I'd like to acknowledge support from a number of different sources over the years. Of course, critical to our work with venoms. And of course, colleagues, including several people that you'll hear from later on in this conference, have been central to our progress in this area, as well as students. And I've been very fortunate to have a number of very capable students over the years. So with that, I'd like to thank you for your attention. And if I have any time, I'd be happy to answer any questions.